Thank you. Okay, I'm just going to... Okay, so this topic of extracellular vesicles is very exciting and developing very quickly, and many people are still not very familiar with it. So my ugly cartoon here really tries to depict what I mean by extracellular vesicles. So of course we think of cells, and we look at pictures of cells in textbooks. They are these beautiful entities. They have a membrane, they have an inside and outside, and it's all very tidy. The reality is that cells are very messy, and they produce structures, various forms of structures, tubes, particulates, and also these vesicle things. And by vesicle, I mean that it has to have a kind of membranous entity here that separates an inside from the outside environment. And there's lots of different types of vesicles that a cell can make. Really in the field, we consider these as two main categories. The first category are vesicles which arise from the plasma membrane. So this is simply a folding, a budding outward, and a pinching off. So the membrane here represents plasma membrane, and there's a bit of cytosol in here. And we would call these microvesicles traditionally. Some of these things are really big, but also, and perhaps inconvenient for us, some of them are really very small. This name of microvesicles there, therefore, isn't very suitable. Perhaps the more correct term is the old-fashioned term ectosomes. These describe vesicles made by certain forms of activated neutrophils, for example. And it's not size dependent as a descriptor. The other major class of vesicle are those which are pre-made, if you like, inside the cell. So there are certain classes of endocytic compartments termed multivesicular bodies or multivesicular endosomes. There are decision-making compartments in the cell, really, and the cargo of a MVB can shuttle the content to a lysosome for degradation, or indeed this whole compartment can be transported to the plasma membrane where fusion events happen. And we have this exocytosis route where these pre-made things are expelled into the pericellular space. And there are other exotic things that cells can produce. So there's a lovely example here of an odd thing called a microsome. There's not much literature on these, but you see these as akin to a multivesicular endosome on a stalk that's kind of snapped off. So certain cells that migrate leave these little footprints, if you like, behind. And apparently myeloid cells can come along and pick these up and gain instructions from this. And in cancer, we get these very odd structures which are produced by some cells called oncosomes. These are absolutely huge things. Again, there's only a handful of groups that are really digging into and studying these things. So most of us really are concerned with these small vesicles, either of plasma membrane origin or of endocytic origin. And the whole thing is messy because cells will also undergo death. Vesicles and debris related to dead cells or dying cells also adds to the complexity and the diversity of the system here. So these are not random structures. These are organized, regulated structures that cells produce both in steady state conditions, when they are activated, when they are undergoing death. And there's a huge repertoire and diversity of structures which are present all the time. All of these extracellular vesicles. So people who've only just heard of these things think that they are, newly discovered entities. And I get really pompous and on my high horse and say, well, actually, no. We've known about the existence of vesicular things for a long time. So the first reports of these things were way back in the 1940s. This famous paper by West and Chandgraf described what they term as platelet dust. So stuff that comes out of platelets, they accelerate the coagulation cascade and help clotting. And then in the 1960s, I guess, when the electron microscopy systems were being developed and people were looking at all types of different biological tissues. Certainly in bone, we see these characteristic dots. These are small vesicles. They're called matrix vesicles. And they act as a nidus, if you like, for mineralization. and allow that tissue to harden up. And then a little bit later in the 1980s, people studying the differentiation of very large reticulocytes that become ultimately these really super compact erythrocytes that we know so well. And how does that process happen? How do these huge things lose their cell mass? And also, how do they get rid of all of this transferrin receptor, which are plenty abundant in the reticulocyte, but absent in the red blood cell? And Bruce Johnson and Phil Stahl also using EM technology there. And this is a classical picture for vesicle researchers, where you see this exocytic cup here, with these preformed vesicles, loads of them, labeled very heavily here with antibody against transferrin receptor. And this is being expelled out of the cell. So some fantastic old school biochemistry was done on these things. The trouble is that they coined this term as cellular garbage, which is really unfortunate, because everybody's interested in the cells and the processes of differentiation, but nobody cares much about cellular garbage, right? We don't rummage routinely through our bins investigating what's there. So that's a really unfortunate term here. This really hasn't helped the field very well in terms of its progression. And it's taken something really amazing, I think, to set the ball really flying in terms of the vesicle research area. And for me, I mean, this is just my opinion. This came from an amazing paper from Graça Raposo. It's a JexMed paper in 1996. I know it well. And she's studying B cells and their antigen-presenting cell function. And she's studying MHC molecules and their trafficking in cells. And we would naturally assume MHC class 2 molecule is a cell surface molecule, but it's not. Most of it resides in these multivesicular endosomes. It's a compartment for loading peptides onto these molecules. And she discovered that these small vesicles are expelled by B cells stained heavily here with antibodies against MHC class 2. So by coating these vesicles with peptides that bind into the peptide binding groove, she was able to stimulate T cells using vesicles, not whole cells, but the vesicles. And these T cells were activated and proliferated and made cytokines. So this is an amazing discovery. So now we don't have cellular garbage. Now we have surrogates of cells with biological activity. And this really launched a massive amount of work in terms of studying vesicles and their immune modulatory capacities. The other major breakthrough came around the same time, really. So Jan Lottwall in Gothenburg in Sweden, he was the first really to, not strictly true, but the first to take the credit for identifying RNA inside the lumen of vesicles. And he was the first to show horizontal transfer of the RNA. So mouse mast cells would pass on mouse RNA to human recipient cells, for example, and the human cells now are making mouse proteins. That's an amazing discovery. Almost akin to a virus hijacking a cell, right? And around the same time, Johan Skog, who's based in Sweden, and then soon after zipped over to Sandra Brakenfield's lab in Harvard. They were studying biofluids and particulate matter in biofluids, and vesicles were discovered to be there in the fluids, and vesicles were in the fluids of brain cancer patients carrying mutated proteins from brain cancer in the blood, for example. And again, that opens a whole repertoire of activity around vesicle biomarkers. And currently, all of this is still evolving, and a mechanistic understanding of how vesicles operate still evolves. There's a huge drive now to maybe manipulate and use vesicles for therapeutic use. There's many companies around the world popping up all the time using stem cell-derived vesicles, either as drug delivery agents, which are poorly defined and poorly understood, or stem cell vesicles in their own right with bioactive effects on tissues and regeneration, for example. So there's a lot of activity on the area now. So it's an exciting and well evolved and well developing field. And number 3 here is where I got involved, of course. Look how well the number of publications have exponentially grown since I got involved in the field. I think I got away with that one. Okay, so bless him, Jan Notval, we were all in a room in Paris, about 200 of us, and we decided to launch our own society, and we all voted that Jan should be the president of the society and take it on. He wasn't in the room at the time, he'd gone back home to Sweden, but bless him, he took this on in good faith. So the concept of having an international society was evolved into this wonderful and mature society that we have now. So of course, it's global society. We have chapters in America, in Europe, and Australasia. And the society holds these big annual conferences. We hold smaller workshops to discuss specific topic areas. As an example here of a workshop I was able to host in the UK surrounding handling blood for vesicle analysis. We release position papers to help guide researchers in the field on best practice and do's and don'ts. There's a position paper here very recently on urine vesicles. This is a big position paper here on handling vesicles and defining them, measuring them, and do's and don'ts. There's about 300 authors on here. These are very influential statements made by the society, really, to help guide and grow the field. The society also has a sister journal, the Journal of Extracellular Vesicles. I remember when this was born, I was an associate editor on this very early on. I remember actually celebrating the end of the year when we had 10 papers published in this brand new journal. This was, wow, this is an amazing achievement. Well, this journal is now massive, and they're barely able to cope with the submissions to the journal at the moment. And the current impact factor for this journal is crazy. It's 25.84 this year, which is absolutely insane. It's just amazes me that where it came from, having, really trying to persuade people to submit things to the journal to where it is now. It's an amazing transition in such a short time. We have national societies now. There's a UK one, a German, a Dutch one, and so on. And we were just about to get into sending postdocs around and exchange visits that we were funds from the society when COVID hit. So really, really bad timing. So none of that has happened, but the plans are for that to continue as soon as it's safe to do so. However, ISAV wants to control these individual societies and come under a European umbrella, if you like, and control what we do, when we do, and how we do it, and have this ISAV branding. So there's a bit of a pushback here now, because I don't think any of the national societies want to lose the autonomy and freedom to organize things that they want to organize and get things done. So this pushback, I think, is all part of healthy debate and how we organize the topic area globally, and how we remain connected and allow collaborative things to happen freely. So I think that will be probably the position that we take in the UK, and I've also spoken to the French guys, and they're going to push back too. So I don't think ISAF's ambition here is going to work out, but that's fine. Okay, so I'm going to jump into the lab then and talk to you a little bit about the vesicles. I've called them exosomes here, but as you know, exosomes are endocytically derived things. And the honest truth is that it's very difficult to be certain that I have exosomes in my hands or I have small plasma membrane derived vesicles. Hence this term extracellular vesicles is non-specific and vague, but it's the best we can do at the moment. So this is a typical lab workflow. So you have a source of material. This could be an ordinary two-dimensional cell culture. It could be organoid, it could be explant cultures. It could even be a bit of tumor tissue that you chop up, homogenate, and then put through a process of vesicle isolation. The process of isolation really should be as simple or as complex as required, as demanded by the complexity of the source material. So a tissue source would require a very hard and stringent multi-step process for isolating vesicles. This thing here is a bioreactor flask. We've used these extensively. It allows us to grow cells at very, very high density in a small volume. So we have tens of millions of cells here in a maybe 10 or 15 mil volume. So the secretome here is already enriched in vesicles. And we milk these flasks every week. And the cell condition medium come off that look like pea soup. So it's complex, it's full of stuff. So we need to pre-clear the debris and the cellular material from that. And then we spin really hard. This step here, the sucrose cushion step, is vital for us in that we're separating vesicles based on their biophysical property of density. So the Grasser Reposo paper I talked about earlier was seminal in that it really detailed some of the biophysical properties of the vesicles in B cells. So having a density between 1.1 and 1.2 is a classical exosome density, and the cushion is very effective at capturing vesicles that are less than 1.2. We collect the cushion and we wash it. There are more elaborate protocols on this available, and there are also simpler protocols than this available. And the essence is, the more complex your protocol, probably the better the purity of your specimen is, but you probably got less stuff to play with. And all of this is time and energy and effort, right? So there's a balance to be had between what's tractable and what's possible. This is a good balance, I think, because we have on a nanoparticle tracking analysis profile, a repertoire of largely small vesicles. They're not entirely monodispersed. We have a range of vesicle sizes here, but most of them would fit that exosome categorization of 30 to 150 or so nanometers in diameter. This graph here is a glorified dot plot, really, where we're immobilizing vesicles on a plate and we're labeling them with antibodies with a high sensitivity reader here. This is a europium labeled antibody and time-resolved fluorescence reader. And it shows these characteristic features of tetraspanins and integrins correctly orientated on the outside of the vesicle membrane. If we had a prep full of contaminating protein, for example, the way we load these plates, it would mean that we lose the intensity of signal here. So it tells us about the molecular profile of the surface of vesicle, but it also tells us a lot about the purity of the preps. These Western blot panels are fairly typical in the field. We compare cells with vesicles which come from those cells. And it shows typical features like an enrichment of proteins related to the endolysosomal pathway. So TSG and ALEX are accessory proteins to the ESCO machinery. So this is multi-molecular machinery grabbing hold of components to dump into the multivesicular endosomes. So we see these are concentrated in our vesicle preps. Well, conversely, GRP94, this is an ER marker. The ER is not well represented in vesicles. We hardly see any bands here, where it's much more present and prevalent in the cell. Mitochondria proteins, nuclear proteins, and other components of the cell usually are not well reflected in the small vesicle. But seeing is always believing, and that's a Grasser Reposo comment. And I think it's really important to look at the specimen for structures, right? Because we are talking about vesicles here and not aggregates or linear membrane fragments, all these other things that might explain the molecular entities that we find in our sample. We could do some traditional TEM where fixatives and negative stains are used, but these give you ugly bloated structures that absolutely do not well reflect the genuine native structure of the vesicle. And in fact, in some of the older literature, you see exosome described as cup-shaped vesicles. They're not cup-shaped, that's nonsense. The cup shape arises if you dehydrate this structure and it implodes to give you a kind of U-shaped structure. With more refined technologies now, and this is fully fledged cryo-electron microscopy here. The trick is in vitrification. So we use a robot. It's a ridiculous device. It's a robot that plunges your sample into ethane, which is pre-cooled to liquid nitrogen. And the device allows you to control the timing and the humidity and all of that stuff to get the optimal ice buildup around the sample. So what we're able to see now are these beautiful native structures of the vesicles. They are spherical. They have this lipid membranous entity here. You can see the inside, you can see the outside. Wonderful. The reality is it's not wonderful because we have other things in there. We have examples here of these bilamellar vesicles, membrane within a membrane. There's another example here of an odd structure with lots of membranous content here. This is a great big ugly thing here. I'm not happy we've got this in our sample, but we have. And when I grabbed this for the slide presentation, I hadn't noticed this before, but this damn thing here is budding off and making vesicles. So these big things can make small vesicles. And we have these other things in the specimen. So these are probably exomeres. They are particulate material that don't really have a membrane. They're very, very small. Nonetheless, they are structural entities and they are present in the sample, even though we've gone out of a way to try and float these and capture small vesicles based on their density. So it's very rare even in JAV papers to see a wart and all descriptor of the product like this. The good news for this is that the vast majority of these things would be the classical exosome type of vesicle, the small, the unilaminar entities. And if you look at things like, I don't know, the volume inside or the membrane surface area, although these things are very small, there's lots and lots and lots of them. So this gives you a big, big, big membrane surface area, right? And that must be relevant for their functionality. So if nothing else, the take-home message today, and I'm going to go on and on about this, is membrane, membrane, membrane. If you're looking at luminal content, remember I talked about RNA delivery, RNA lives in here. Really for big lumen and volume, these things are really giving us most of the volume here, even though these are rare events. So membrane, membrane is the mantra of the day. I grabbed this, this diagram from Google. I was just looking at a way of describing the structure of a vesicle from a molecular perspective. And look at that. I mean, it reminds me of some of those 3D images of viruses that you see. Viruses are much, much simpler. They're defined, they're reproducible, they're uniform and predictable. Well, this is mayhem. Look at all the different types of membrane proteins that are attached on this. This membrane is incredibly dense with transmembrane and GPI anchored proteins. And in fact, as depicted in this diagram, I'm not sure whether I agree that some of these things are present on the membrane and whether they are present inside, or whether in fact they are not oriented as they're shown in this diagram, or whether they are reversed inside. So the topology of the arrangement of the molecular content of the vesicles is actually quite controversial. And there are examples where you see MHC class 1 here is absolutely oriented in the right way, but you get examples of some proteins which are upside down, and we don't really understand how that happens. And we also have to consider the lumen here. It's full of a cytosol-like environment, but there's examples where specific sequences of RNA, for example, are grabbed hold of and are enriched here. So if you profile vesicular RNA compared to cellular RNA, they look very, very different. Most of the vesicular RNA is non-coding. There's very, very few transcripts in there. It's as though it's junk material, perhaps, that the cell is sending out. Uh-oh, garbage again. Okay, so cancer cells are making these things all the time. Other cells in the environment will also make these things all the time, and they go off into the pericellular space, and they will hopefully interact with some kind of recipient cell. And how might that interaction take place, and how does it happen, and what's the consequence of that? So the simplest type of interaction we could imagine is a vesicle binding to a vesicle receptor of some kind and instigating the signaling response in the recipient cell. But given that cartoon I've just shown you with that complexity of membrane, probably what we're actually going to be seeing is a multiple delivery of a host of things, adhesion molecule and other things, signaling perhaps parallel responses in the recipient cell. And what happens to that vesicle after? Does that signaling last forever and a day, or is it dealt with? Well, probably the cell is dealing with that in some fashion, switching things off, and it has to deal with that particular entity on the membrane. So it's going to be drawing that thing inside the cell, probably through an endocytic process, and processing that material. Maybe that will also give us some signaling responses as that happens. This is a quirky thing that we published a while ago, actually, where vesicles can influence the recipient cell without actually coming into contact with it, which is perhaps counterintuitive. But membrane, remember. So we have membrane enzymes, don't we? So ATP and ADP can be digested by these enzymes in the membrane. So we get a loss of substrate and the gain of a product, which can then go off and stimulate through signaling receptors and so on. The other good example of this is arginase driving arginine consumption, and that again has an impact on endothelial cells and T cells, I believe. So what are we going to study in terms of our recipient cell in these experiments? So epithelial cells are here, they're making lots of vesicles. The vesicles are going to interact pretty quickly with stromal cells, right, which are immediately in the vicinity. And the stromal cells seems like a pretty good thing to study in cancer. This is a review from 2006, and you may or may not agree, but certainly the authors of this review put the stromal cell at the center of the tumor environment in orchestrating changes and really calling the shots in terms of what happens, producing factors that can influence angiogenic responses, factors that can chew up and change matrix, redirecting matrix, the type of matrix that is deposited, making chemoattractants, controlling infiltrates, making epithelial cells more motile and aggressive and invasive. So all of these things can be directed by the stromal cell. And in prostate cancer, we see very dramatic stromal cell alterations that occur very early on, even in precancerous lesions. In this example, with these beautiful, juicy, smooth muscle cells here in a healthy prostate, and this becomes a more fibrotic, collagenous type of matrix environment very soon with the onset of prostate cancer. And if you stain the cells in this interstitium with antibodies against alpha, smooth muscle actin, and vimentin, it shines up brightly. So these are classical myofibroblasts that seem to appear in this interstitium. I know this is a small study, but in general, if you have lots and lots of this odd stroma, these patients do poorly. And if there's not much reactive stroma in the tissue, the patients do much better. This is a very dramatic example of that in a xenograft. A very simple experiment. We have an epithelial cell as the tumor cell, and you implant that with fibroblasts from benign prostates. So these are men with enlarged prostates. They're not cancerous. This is quite common in men over 50. Or you can take stromal cells from men with prostate cancer. You put them into mice with these epithelial cells, and look at the dramatic difference here. These massive bloody tumors arising. And the only difference here is in the phenotype of the stromal cell. By bloody tumor, I mean, of course, this is red and very well vascularized. So the host vasculature is being co-opted in terms of supporting the disease here. So the stromal cell has a really profound effect on the outcome. So how do we get myofibroblasts in the tumor? Well, they probably arise from a mixture of all of these things, really. So even when we think of a terminally differentiated specialist luminal epithelial cell, under the right situation, these can become mesenchymal-like through that process of EMT. There's also a story where blood mesenchymal stem cells may be part of the infiltrate and get into the tissue and take hold as these myofibroblastic cells. But the easiest thing to study really is to take a fibroblast from skin or lung or big toe. You can stimulate these very simply with TGF-beta, and within 3 days, you see this completely dramatic transformation in the phenotype of the stromal cell. So they become alpha smooth muscle actin positive, but that's not enough. This is actually polymerized into these stress fibers. So these cells have now acquired a contractile phenotype. And if you're in a skin wound, that's perfect, right? Because you now have the capacity to close the size of those wounds with this contractile activity. But these cells persist in solid tumors. They seem not to resolve, and they secrete all these factors that can orchestrate tissue rearrangement and promote progression. So in the early days when I was in Valendra, we were throwing vesicles on blood cells and seeing what happened. And we saw that T cells were very strongly suppressed by tumor vesicles. And we were trying to work out how they do that. One of the ways to do that is that the tumor vesicles carry TGF-beta. So this is a very elaborate way of proving that, really. So we had these analytical gradients where we have sugar at low concentration or high concentration, and we load vesicle concentrates at the bottom or at the top, and we spin these tubes really, really hard overnight at 210,000 G. And the vesicles will move along here and stop when they reach their equilibrium density. So you collect zero fractions from here, you measure the density by refractometry, and then you can measure the fractions for where the vesicles are. So they're here where this TSG 101 is between its classical density range here, between 1.1 and 1.2 grams per mil. And the blue bars here represent TGF-beta ELISA. So we have a soluble form of TGF-beta produced by the cell, but we also have a tightly vesicular associated form of this growth factor 2. Different tumor cells load different amounts of TGF-beta on their vesicles, and when we add this to a luciferase reporter assay, so this is a way of measuring TGF-beta functionality, this graph and this graph look exactly the same. So we have functional TGF-beta being delivered to recipient fibroblasts in the form of a vesicle. So we wanted to take this a little bit closer to the patient, and we were really keen to see whether we could get hold of stromal material from men with prostate cancer. So our surgical colleagues in Cardiff Vale and the Wales Cancer Bank have been really fantastic in helping us get these collections up and running. So men with prostate cancer, those who have fairly high-risk disease where the tumor is at risk of getting out of the organ and into the pelvis or into the nodes. Some of those men elect to have their prostates removed. So this is the bladder, this is the prostate, this is where you pee through. And the prostatectomies go to pathology, and we ask for disease that was unilateral, present in one side of the prostate and not the other. And we ask for the needle to go through that tumor, and we'll have a bit of this core. And again, the needle to go through a bit of the normal, that's normal with inverted comma, side of the prostate. The whole organ is, of course, disease. So we don't know whether these stromal cells actually represent normal or slightly abnormal cells. We put them in culture, and by passage 2, we've cleaned them up. We've selected out the epithelial cells, which are frankly impossible to grow anyway. And we have these fibroblasts growing in culture nicely. The normal cells are fibroblasts. The disease sites are a mixture of fibroblasts and alpha smooth muscle actin positive myofibroblasts. So we took these normal stromal cells and we stimulated them with TGF-beta or vesicles, and we looked at the nature of the myofibroblasts that arose 3 days later. And there's a huge range of difference between the TGF-beta cell and the vesicle-stimulated cell. All of these growth factors are made differentially. The really sentinel one for me is HGF. So HGF is secreted shed load by tumor-educated stromal cells, or also by our vesicle-educated cells. The TGF-beta variant here, actually, we see a loss in TGF-beta. So if you were to compare these myofibroblastic phenotypes, our vesicle stimulus, it's a one-off stimulus with vesicles. We're getting a myofibroblast here, which is really similar to the cells that we're extracting from tumors. This phenotype also is true when we look at function. So these are the TGF-beta generated myofibroblast monolayers, and we dot endothelial cells on top of these monolayers and let them sit there and see if they take. You can see a dramatic difference here. So this is not an endothelial cell surviving environment here. Whereas with the vesicle-generated myofibroblasts, these endothelial cells survive, they move, they get organized into these branched vessel-like structures. So this is quite a pro-angiogenic environment. In mice, if we xenograft tumors alone, they will grow. If we also add myofibroblasts generated by the vesicles, we get this heightened growth here, akin to that bloody tumor that I showed you earlier. This is weird. The TGF-beta variant of this is actually controlling the tumor growth in the mice. So these are completely polar opposite type of myofibroblasts that we're seeing here. And certainly the vesicle-generated form is much more akin to what we see in terms of disease-educated stromal cells. This soluble thing, it looks quite artificial. I mean, it really begs the question how relevant these soluble factors really are. and was everything really delivered by vesicles. So clearly the phenotype here is so different between a soluble classical stimulus and a vesicle stimulus. This simple receptor ligand signaling thing can't be true. There's got to be more to it than that in order to fine-tune the fibroblast response that arises. So maybe we have a multiplicity of events happening at the cell surface, giving us multiple signaling cascades in parallel. So when we were scratching our heads, thinking about how TGF is tethered to the vesicle surface, beta-glucan was a really obvious candidate because another name for that is TGF-beta receptor 3. It's a non-signaling receptor. It binds TGF-beta with low affinity, but perhaps high capacity. And this little trick of treating vesicles with pivanidate, removing an inhibitor, liberating the proteolytic activity of this MMP, can cleave beta-glucan from the vesicle surface. And this worked quite well. So we lost beta-glucan and we didn't lose other things. We lost TGF-beta and we attenuated the differentiation potency. So that's great. So we have TGF-beta associated to vesicles via beta-glucan. Another way of playing with this system is that beta-glucan is a heparan sulfate proteoglycan. So it has these side chains of heparan sulfate. So we got an enzyme in, which is quite expensive actually, because it has to be certified protease free. This enzyme is selective and it will chew these side chains off and leave the proteins intact. And we have an antibody specific against HS side chains, or an antibody specific against the little stubs which are left after digestion. So the graph shows that the digestion works really well. We've lost our heparan sulfate side chains, but we haven't attenuated other surface proteins. And the Western here shows the absence of these stubs when we use enzyme that's been heat inactivated. And we see multiple bands here in terms of different core proteins, I think, when we liberate and reveal those stubs for the delta HS antibody to bind. This big thing here is probably beta-glucan, but there are other things here as well, which are HS chain positive. Now, when we've shaved the vesicles, I was really surprised actually. We didn't see any loss at all in the total amount of TGF-beta tethered to the vesicle surface. But now when we add these to our recipient fibroblasts, we've really strongly attenuated their ability to drive differentiation. And this is no longer a pro-angiogenic environment. So we've almost entirely lost the functionality of these cells by stripping the HS side chains. We still don't really understand how that works, whether there's a molecular handover mechanism here where the side chains are necessary for efficient handover to the signaling receptors, or whether there are a whole bunch of other factors which are tethered to these HS side chains, which are participating in driving the nuance of that myofibroblast phenotype. So this was a really interesting question, and we had a PhD, Stephen Sarah Vega, who actually passed Survivor about 2 weeks ago, studying this with us. And the enzyme trick was extremely important for her project. So we chopping these things off with the enzyme. But we also tried another approach, which was to down-regulate the co-proteins using an shRNA system. The graph here shows vesicles isolated from those modified cells carrying the knockdown, showing reasonable attenuation of co-protein on the vesicles. I was a bit disappointed actually about the strength of attenuation here, but we have at least 60 and sometimes up to 70% loss of these co-proteins on the vesicles. So what we're looking to do is profile the vesicles to see what other growth factors are on them other than TGF-beta, if there are any at all, and then to explore the role of HSPGs in tethering those growth factors to the vesicle surface. This is going to be quite challenging because there's not going to be a lot of growth factor around. We don't have many vesicles to play with. So we had to employ something very sensitive here as the readout. So we used a proximity ligation assay to do this. These are quite clever little assays in that you have an analyte here, let's say interleukin-1. You need 2 antibodies against interleukin-1 binding to distinct epitopes on the analyte. If they are near enough to each other, these short oligonucleotides will hybridize and form a short bit of double-stranded DNA here, which you can then PCR amplify out. So we had 3 panels encompassing around 270 or so analytes in this experiment. So my expectation is we shave the vesicles of the HS side chains, and we should see a whole bunch of things fall off. And that's exactly what we saw. So we saw about 40 or so analytes that were now no longer strongly expressed on the vesicles. These include some chemokines such as MCP1, some inflammatory factors, some interleukins, midikine and C61 were very abundant. So we can now say that the vesicles host a whole repertoire of these cytokines and chemokines, and a great many of them are tethered to the vesicle surface by association to these charged heparan sulfate side chains. We confirmed some of these things by ELISA. When we did the other route of looking at this, knocking down a core protein, so this would represent glypican-1 deficient vesicles, glypican-6 deficient vesicles, beta-glycan, synuclein-4 and synuclein-3. I was really quite surprised and semi-horrified by the complexity of what came out here. So instead of losing stuff, when we knock down glypican-6, for example, we've gained a whole complex repertoire of growth factors on the vesicle surface here. And we still are far from understanding exactly how this works. There is an emerging story where, as vesicle biogenesis happens in endosomes, these syndicans and glypicans are fairly important in dictating how it's made, and what gets hooked in and what gets dumped onto the vesicle before it's secreted. And I think this experiment shows us that we really don't understand vesicle biogenesis very well at all. But it certainly highlights the potential relevance of these elements on the vesicle surface in terms of how they work, how they're made, and how they deliver growth factors to recipients. So if I go back to the simpler system now, the vesicle shaving, if we stimulate fibroblasts with the vesicles, we see a whole range of phosphotyrosine receptors activated here. We kind of expected that, right? But this is telling us we have some element of parallel signaling going on. And again, with the attenuated shaved vesicles, many of these pathways are now no longer activated. So we lose the efferent signaling. There's a FLT3 thing here that we completely lose. And perhaps oddly, we have, what is it? We have this HGF receptor signaling that is actually reversed. So it's actually enhanced when we do the shaving. So the cellular response to the vesicles is highly complex, but it clearly shows us we have multiple signaling happening at the same time, and that HSPG side chains are really relevant for most of these things that get switched on. The cytokine output from vesicle-activated cells is also complex. And again, if we shave the vesicles, we're seeing really strong attenuation of some of these things, which are no longer made or now made much more weakly. Or in fact, if we take regulators away, we're now activating some things that were not there before. So this is a really, really dynamic and hyper-complex response of a cell to a vesicle population. And HSPGs are really sentinel really in fine-tuning the fibroblast response that arises.
Just to mention, Aled, it's coming up to 10:02. You asked me to let you know.
Okay, thank you, Arwen. I'm going to zip through the endocytosis, but I apologize, Arwen. But we've also looked at entry following binding. This is a very complex thing to do. There's lots of ways of fluorescently tagging vesicles. We've done it largely by fluorescently tagging the protein shield around exosomes. This works very well as a labeling strategy, and it does not interfere with differentiation function. So vesicles bind to cells, and they gain entry if it's warm. It seems to be a saturatable thing. Most of them go in by 2 hours. This is an image of a highly saturated dosing scheme here. So we tiered that down to a dose that's more manageable to help us look at differences. So Arwen's designed some really lovely siRNAs that are very specific to endocytic pathways. And when we block this AP2 thing, which is a clathrin-dependent regulator, we see really nice attenuation of vesicle entry into fibroblasts. Some of the pharmacological inhibitors also work. Dinosaur works really well, which would be consistent with the clathrin-dependent attenuation. Vesicles get into cells, they move, and they move through this classical route really through early, late endosomes through to lysosomes. So after about 4 hours now, we see co-localization of our vesicles with dextran-loaded lysosomes. And this is a classical route. Presumably they get degraded in the lysosomes. We've also asked the question, what happens, however, to the lumen? So we've tried to label the lumen again with fluorescent agents. We've tried a lot of things. Most of them were a bit problematic. Cyto-RNA Select worked really well for us. So this is an RNA labeling dye. It's fluorescence when it's in complex with RNA, and it doesn't bind to DNA. So we can inhibit EV delivery of this dye using Dinosaur. And there's reasonable co-localization between this RNA dye and a maleimide protein coat when the vesicles are outside the cell. When we add them to the cell, we see something quite weird in that although we see the occasional puncture here representing endosomes, we also see the stringy distribution of this EV cyto dye. Pete Watson said this looks like mitochondrial labeling. So we threw in some mitotracker here, and I think he was right. So most of this seems to be co-localizing with mitochondria. If we label vesicles with protein and also co-label with mitochondrial dye, we see no co-localization at all. So the proteins are not going to the mitochondria, but the lumen seems to be. Now, if we label the cells with vesicles, which have this cyto dye in, or indeed the protein coat are labeled too, we can see that outside the cell there was good co-localization, but within an hour of being in the cell, these two signals are pulled apart. So the vesicle protein is going to the lysosome, and the luminal content, the RNA, seems to be going to mitochondria. We had a few sessions actually at this very, very new beamline at the Diamond Synchrotron facility. So this is a soft X-ray microscope. It's a cryo soft X-ray microscope. It's under vacuum. It's all at liquid nitrogen temperature, and it's an absolute nightmare. This is the Vitrobot here. So what we've done, we've labeled fibroblasts with the vesicles. We've waited for a time, and then we've looked at ultrastructure, at where these things go. So this is the cell nucleus. The green here represents this cyto-RNA dye delivered to the cells. And this gray section here shows us the ultrastructure in this region. So we see these sausages here, which have labeled M, representing mitochondria, and these open round structures here, which have labeled E for endosomes, but they may or may not be endosomes. The point is there's a lot of these things around in adjacent regions to these mitochondrial structures, and it opens a possibility of endosomes receiving extracellular vesicles and directing the endosomal escape towards a mitochondrial border here. We see these zones of high membrane area at the contact points here. We haven't proven that. That's a really quirky idea. We seem to be getting delivery of RNA into the mitochondria, but there's a bit of work to be done on that. So tumor cells make vesicles. They're very diverse structures. There's big things, there's small things, there's everything in between. Molecularly, they are highly complex. They can drive a program of differentiation of stromal cells. We've used fibroblasts. We've also used mesenchymal stem cells to show this. The phenotype is similar to that which we see from tumor-educated stromal cells. So they're much more physiologically relevant than maybe treating cells with soluble entities. Heparan sulfate proteoglycans are really critical in how the vesicles operate and deliver a concert of soluble factors to the recipient cell and direct that signaling. They endocytose very rapidly and very efficiently by the fibroblast. The protein gets to the lysosomes. They actually stay in the lysosomes for a long, long time, 3 days at least. That might be a property of our labeling, but we've also labeled vesicles in different ways with GFP and other systems, and they are very longly retained in the stromal cells, whereas degraded very rapidly by other cells like epithelial cells. We don't understand that very well. And there's this weird phenomenon and observation where the RNA seems to be delivered into the mitochondria, which is really quirky. I love quirky. It's a puzzle. I don't understand it. And we've got lots more work to do on working that out. Right, before I finish, I just like to touch on this briefly. So vesicles are in biofluids, and maybe we can measure them, and maybe we can get some intel on the status of disease by looking at the content of these vesicles. Joe Welton was a postdoc in my lab, and we developed some protocols. I mean, these are ridiculously complex, right? So I know that they're crazy. They're completely intractable. But what they do is allow us to isolate vesicles from very complex source material. So this is a plasma sample. And guess what? We have small vesicles of different structural types as well as some amorphous material. They're small. They have these tetraspanins. We've got rid of the albumin. In urine, it's slightly easier and even cleaner. But again, we see this diversity of small structures. We have these classical endolysosomal markers here. We cannot get rid of this damned uromodulin stuff, this tam-horse protein, so we live with that. So in our hands, we have this repertoire of patient-derived stromal cells, and we have the normal and the disease matched. No one's ever looked at vesicles from these cells, so this is something we've started doing. This slide just shows a very quick characterization of that according to the ICEV guidelines. If we isolate these vesicles and attack them with RNAs, we do not lose the RNA there. So the RNA resides encapsulated and protected from the harsh outside environment. And we can extract this RNA and we can run a bunch of PCRs on them. So this is a low density PCR array. And we look for obvious things, stromal related things, fibrosis and inflammatory type of things. And we saw a whole bunch of difference between normal stroma cells and disease stroma cells, and equally between the vesicles from those are also different. So we had a handful of targets that we looked at all of our patients, and some of these were consistently up and some of these were consistently down. So fortune favors the brave. And I did not think for 1 minute that this would work. So we got some serum samples from the World Cancer Bank. These are in two cohorts. The first set are from men with cancer, but this is clinically insignificant disease. So the histological severity here is not a real concern. So these men would be monitored. And the other sample is clinically significant disease. So for many of these, we have grade 4, grade 5 disease. These are of concern. So these are the guys who might need treating. So it might be really great if we had an assay to help us discriminate group 1 from group 2. So we ran a whole bunch of PCRs, our targets on these vesicles isolated from these serum samples. And I guess, as we might expect, most of them didn't hold true and were not useful. But we had the odd one that looked quite useful. So we went and knocked on the door of Peter Giles in the Wales Gene Park and said, look, we have these multiple measurements. Can we combine them somehow? How do we do this? Can you help us? So yes, so let's do, artificial intelligence, machine learning is the thing of the day. So Peter is really great and really flexible and creative in doing these decision tree things for us. And of all the candidates, these 4 or 5 popped out as being extremely useful and potentially discriminating clinically insignificant from clinically significant disease. So we ran these ROC curves, even though with this small sample set, so bear that in mind. So PSA alone as a measure to discriminate low versus high glycemia was quite poor. Our RNA combined scores was better. Combining our RNA scores with PSA, because we have that measurement anyway, was really great as an assay in terms of that discrimination between low and high Gleason score. And of course, rule number 3 was absolutely right. Your sample set is small. Is this real? Can you reproduce it? So we did it again on a different cohort of samples, and the same thing has held true. These numbers are much more real world and more believable, I think. But the pattern is that our RNA is better than the PSA alone, and our RNA plus PSA as a combined score is better still, identifying significant from insignificant disease. So this is a very ropey assay at the moment, and we have an ongoing project to look at things in the vesicles by RNA-seq, which are less obvious. The proportion of messenger RNA in a vesicle is as low as 4%. Most of it is non-coding. So we're really digging into the non-coding elements by RNA-seq now to discover elements that will better discriminate the disease from the normal stroma and see whether we could pick up these signals in blood in this way. So before I go, I'd really like to thank colleagues at the tumor microenvironment group, especially Jason, who's been instrumental really in the stroma work and some of the recent RNA work. But all of these people roll their sleeves up and get stuck in and involved in the study. Huge thanks to Peter Giles for the AI work. Huge thanks to the Wales Cancer Bank team and our urology colleagues at Cardiff and Vale, and to Arwin, Pete, and former PhD student Alex for the endosome study, which I zipped through really quickly. So I hope I'm on time, Arwin, and I'll stop there.
Lovely. Thank you very much, Aled. So there's, a lot to extracellular vesicles and many different features you could study. That was clear. Thank you very much. So Duncan's asked a question, which is, do micronuclei that occur during periods of genomic instability end up in extracellular vesicles? So I guess, do the vesicles spread the chaos? I don't know if that's what you mean, Duncan, but
yeah,
over to you, Aled.
Yeah, so in the early days, when Laurence, Zietwegel, and Clotilde were setting up clinical trials where they're going to use antigen-presenting cell vesicles as a product to inject into patients as vaccines, right? So one of their criteria, it was an FDA-approved criteria for is it a good prep or is it not, was the absence of DNA. And if they had DNA in the vesicle prep, it would fail their criteria and it wasn't considered good enough and it would be dumped. So from that work, DNA is not actually present in classical vesicles. But since then, there's loads of report now describing fragments of DNA of mitochondrial origin secreted in a membrane-bounded entity, which is protected by this RNA thing. And we have groups in MD Anderson who claim to be able to scoop vesicles from blood and do whole transcriptome profiling from those vesicles and find mutated oncogenes and things in there. So my answer to Duncan is, with your specific question, I don't know. But there's a whole bunch of stuff we don't know about the vesicle field and the exotic vesicles that exist out there. So I think if Duncan, if this is a question you think you'd really be interested in chasing down with us, I'm extremely happy to help investigate that with you.
Brilliant. Okay, thank you, Aled. You two can take that up at another time. So Helen is asking, Helen Pearson, will it be possible to expand your EV biomarker assay to identify oligometastases?
Yes, I don't see why not, Helen. It's all about what you measure. It's the options are too wide. Actually, that's the trouble. So if you profile broadly, you can't do a lot of samples, right? So you narrow your profile down and then you can look at more samples. And then at that point, it becomes more clinically tractable and relevant. So I think the answer is yes, but the design of it and what you actually look for is the problem.
So do you think, Aled, that with looking for EVs as a biomarker, is the main problem sensitivity, the ability to detect cancer EVs over what's in the blood anyway?
Yes, I think so. And there's a lot of noise in the field there, Arwen, as well. It might be that if you have a blood and you do a really crude concentration of that stuff, and you don't care what's in it, but you can measure your marker of interest. Right? You don't care whether it's vesicular, avesicular, you don't care, as long as you can measure it. So there's a lot of this kind of thing out there at the moment. And they say, oh, we've isolated vesicles and therefore vesicles are carrying this marker. From a purist vesicle nerd like me, I'm concerned at that because they haven't defined it as a vesicle entity. It may be just stuff. From a biomarker point of view, you don't care. It's stuff that's measurable and it's informative. So we have a bit of a wrestle there. There are some very well evolved companies out there. One of them is called Exosome Diagnostics. So this is a company that has eventually evolved from Johann Skog's discovery of vesicles in biofluids as markers. And again, they have an interest in the prostate field. They have very elaborate, refined workflows for processing the specimen. But what they found was that if they took any old biospecimen from any biobank, noise, they could not get a signal from that was meaningful. So they created their own biobank and their own cohort in order to profile these things. So when they go to clinic now, you have to ascribe to their SOPs for your specimen collection, or their assays won't work. They're so fussy. Our assays seem to work with ordinary serum from Wales Cancer Bank, whatever their process is. It seems to be tractable enough for that. You don't, if an assay is so fussy, you have to handle a sample in such a fussy way, it's not going to really have a big impact. So it's a very noisy field, the biomarker field, Alwyn, and there's a lot of good in there, and there's a lot of quite bad poor practice and challenges that we face.
Yeah, well, it makes it difficult for the rest of us who are not in the field to sort of navigate through it. So it's, interesting. Helen is also asking, Aled, are HSPGs lost in prostate cancer? And if so, do they correlate with increased reactive stroma and poor outcome?
My honest answer, Helen, is I don't know. Gosh, she asked good questions, didn't she, Helen? I don't know, Helen. I'm going to do some reading. I'll get back to you.
Okay, great, thanks, Aled. So I want to ask another conceptual question. So you're obviously a big fan of the paper that Laurent Sittvergel published on exosomes as antigen presenting cell surrogates. But to me, I can't see how that fits with how the immune system is supposed to work. We want to be able to induce antigen-specific T-cell responses in the right place at the right time. And if you've got these rogue vesicles, well, maybe that you would argue they're not rogue, but they sort of go off, from the big toe where maybe you have your infection and initiate immune responses, in your arm. Then what's the point of that? The whole point of having, lymph nodes and draining lymph nodes and dendritic cells to induce your T-cell responses to make sure that you control the system.
Yes, so, I see exactly what you mean. I mean, these clinical trials only went so far and then they stopped, right? And another French group were playing with tinkering with the fine-tuning of the dendritic cell source in order to make really potent vesicles, and they were really trying to push that. Again, these trials have not got very far. So the vesicles are part of that communication between an antigen-presenting cell and the recipient. They're part of that. But whether they can replace that is another question. And I think the dissemination of vesicles, how they are disseminated from big toe to left arm, these are really fundamental unknowns in the field. It's amazing how much we don't know. And it may be that the capacity to diffuse way past the pericellular space is more limited than we think. And it's only in really severe pathologies where we have this leaky vasculature and tumors, and then you get a more capable dissemination systemically at that point. If you have a tissue that's pretty intact, it may be much more limited and much more localized. Yeah.
I mean, you could imagine a situation where, production of exosomes by tumors might end up creating tolerance via transferring antigens to the wrong places. That somehow seems more feasible to me. But
yeah, and to me too. And that has put us in a bit of loggerheads with the Paris team, actually, because when we were trying to stimulate T cells with vesicles, we could not do it.
Yeah, it's a long time since that paper came out as well.
Yeah.
Okay, so do we have any more questions? No more. So that means it's time to start the weekend.
Excellent. It's beer o'clock.
Fantastic. Thank you ever so much, everybody, for attending, to Aled for presenting. And Aled, could you put the last slide up just so that
I'm so sorry.
that the next webinar is on the 17th of December, so ending with one more just before Christmas, and this is going